Dear students, welcome to Tungal online classes. Today we will solve other level 2 questions, that is NEET level questions now, right? So let us start with the first question now itself, right? Which one of the following is not a constituent of cell membrane? See here, this word that matters a lot, right? So not a constituent of cell membrane. Which is not the constituent of cell membrane? Cell membrane is made up of what? So lipid bilayer itself, right? That is glycolipids are present there, and it is also made up of fatty acid tails, and even consisting of the proteins itself, right? If they are associated with the sugar, so that is called as glycolipids, glycolipids as well as glycoproteins, they are called as, right? All these are the major components of a cell membrane. So let us see the options there. Glycolipids, of course they are present. Phospholipids, the type of lipids are phospholipids, of course they are the constituents. Proline itself is one type of amino acid, so that's why it is not the constituent part of this membrane. So cholesterol is also present there, so that's why the correct answer for this one is proline itself, right? So one thing. Next one, telomere repetitive DNA sequences control the function of eukaryotic chromosomes because they are RNA transcription initiators, help chromosomes pairing, prevent chromosomal loss, act as replicon, right? So here, so analyze the question first and later analyze the options there. So what is the question? Telomere repetitive DNA sequences. So in telomere is nothing but terminal part of the chromosome. It has a repetitive DNA sequences. Control, what it controls? The function of eukaryotic chromosome, right? It controls the function of eukaryotic chromosome because they control the chromosomal function itself. Being as a terminal part of the chromosome, it controls the function. It has a repetitive DNA there, so avoids the loss of chromosome. Why? Because it is tightly coiled there at the terminal part. It avoids the attachment of another chromosome, even the detachment of the terminal part of the chromosome it avoids. Because of this reason, it controls the function over there. So that's why the exact answer for this one is it prevents chromosomal loss. It prevents chromosomal loss is the correct answer for this question. Next one. Here, act as a replicon. Replicon in the sense that copy. It is a replicated part of the copy, right? So this word you must remember over here. So next question. Which organelle present in higher number in secretory cells? So which organelles are more in number in secretory cells usually? The question is very interesting one. So very important question it is. So as you know, the secretory cells mainly consisting of one of the cell organelles like the Golgi complex itself. So that is also called as the dictyosomes in plant cells as well as some of the lower organisms, right? So and these dictyosomes, as they help in packing the materials, and so they are exported, right, through the cell itself. That is the major function of this Golgi complex. Hence, so this is the correct organelle which help in, right? So which is helping in secretion of the materials, being in the secretory cells especially in glandular cells, right? So that is why, so the correct answer for this question is dictyosome. What is the correct answer? Dictyosome. So lysosome, you know, it consisting of many of the hydrolytic enzymes which help in intracellular digestion itself. That's why lysosome is not a correct answer. So endoplasmic reticulum, it has its own functions, it doesn't. So next one is vacuole. Vacuole has totally different function itself. So it contains a cell sap itself, right? It is surrounded by a thin membrane, so which resembles a plasma membrane called as a tonoplast, right? So this is all about the other options, right? Next one. In flagellar membrane, which enzyme catalyzes ATP? Right? In flagellar membrane, as you know the structure of flagella, right? In the flagellar membrane, what happens? Which enzyme catalyzes ATP? Which enzyme catalyzes ATP? Right? So cytoplasmic dynein, axonic dynein, kinases as well as myosin. So myosin is usually present in muscle fibers, right? It is a kind of protein, right? So next one is kinases. It is also a different aspect. Here the axonic dynein is the major, so enzyme catalyzes the ATP, right? So that's why the correct answer for this one is axonic dynein. So it is the protein present in the hook-like side arms, right? As you see the structure of flagella, this is the outer membrane here that you come across. Here at the center there is 2 signets that you come across and a 9 number of doublets that you come across on the peripheral side, right? So all are interconnected over here with the help of interdoublet. So and their side arms that you come across, one is outer and another one is inner. This outer one is looking like a hook-like structure, right? And that is made up of dynein with adipase, right? So that's why here the major answer for this one is axonic dynein. Why? Because so this one is entirely said to be as the axoneme. What it is said to be as? Axoneme. Axoneme it is called as this microtubular structure. What we see over here itself is axoneme, it is called as. Okay, so axonic dynein protein makes this hook like side arm itself, right? So that's why the correct answer for this one is axonic dynein. And the next question is very important. Enzyme found functional in lysosome is. Which enzymes are functional in nature in the lysosomes? Very important question it is. As you know, lysosome consisting of more than 40 varieties of hydrolytic enzymes there. They are acidic in nature, right? Among them, nucleases, which digest the nucleic acids, right? So nucleases, phosphatases, sulfatases are different types of enzymes that we come across inside the lysosome, which help in digestion of all the components of the cells, and even it entirely digests the entire cell itself. Hence, so these are said to be as a bag-like structure which are responsible for the suicide of the cell. Hence, these are called as suicide bags of the cell. Lysosomes are called as suicide bags of the cell. Then, so search for the answer for this question. Enzyme found functional in a lysosome is acid phosphatase, basic phosphatase, oxidoreductase, lyase. Right? These are all different types of enzymes that we come across. But the active one in these lysosomes is acid phosphatase is the correct answer for this question. Next question. The enzyme which increases the rate of permeability across the membrane. The rate of permeability has been enhanced by the enzyme, right? Which enzyme increases the permeability, right? So is major aspect. So here permease is the name of the enzyme. Itself is the name of the enzyme which enhances the permeability of the membrane there, right? So then, which is the correct answer? Permease. Gelatinase is not the answer. Here, catalase, amylase, these are different enzymes. They do not increase or enhance the rate of permeability in case of these. So membranes itself, right? Permease is the enzyme, right? So next question. Lysosomes are formed by. Already I have covered this one in theory. So as the pinched off vesicles of the endoplasmic reticulum makes the tubules of the Golgi complex. On the cis as well, on the cis side of the Golgi complex. So very important point. Cis as well as trans are the two sides of the Golgi complex that you have seen. On the cis side, cisternae, it makes many vesicles of the endoplasmic reticulum together form the cisternaes of the Golgi complex, and later tubules of the Golgi complex. And once again on the trans side, it so releases some of the materials filled vesicles there, and they become lysosome. One of themselves become lysosome. That's why. So these lysosomes basically formed from the Golgi complex, vesicles of Golgi complex. So since it is secretory in nature, so it fills some of the hydrolytic enzymes within that one that forms the lysosome, right? So the correct answer for this one is Golgi complex. So not the endoplasmic reticulum, mitochondria as well as both A and B. Of course, so we can say both A and B also, right? In this case, why? Because so the vesicles of endoplasmic reticulum are also responsible for the formation of lysosomes. Okay, so Golgi complex is the major. Next one, histones are. What these histone proteins are? Where actually these are present? Already you know it, but I shall draw the diagram and you can understand now. Right? See here. So this is one chromosome. Okay, this is made up of the chromatin material itself. As you know, it is made up of highly coiled chromatin material. When you uncoil this one, that moment you will come across the nucleosomes. These are nothing but nucleosomes, which are formed as a result of the histone proteins as well as surrounded DNA itself. Right? DNA as well as histone proteins together form the nucleosome. Hence, this is the model of DNA called as nucleosome model of DNA. It is called as. Here, what happens? DNA is acidic in nature. DNA is acidic in nature, right? That is why it is called as a deoxyribonucleic acid. Its name is. So there, acid is there in its name, of course, because of the presence of one of the component. I do not analyze all those things now. So you haven't studied at the structure of DNA. Whenever you study the structure of DNA, that moment which is the component of the DNA responsible for the acidic nature, that is nothing but phosphoric acid. Now I shall mention, but you do not understand that one. So whenever you study, so the 3D model of DNA, that moment you will understand it. Okay. So here DNA associated with the histone proteins. So if it is acidic in nature, then it has to combine with the basic material. Hence, these histone proteins are basic in nature. Basic proteins these are. They are basic proteins. Hence, the answer for this question is, so histones are the basic proteins. These are the basic proteins. Okay. So next one. Bacterial, gram-positive bacterial cell wall contains. So cell wall mainly consisting of the mucopolypeptides. Yes. Bacterial cell wall contains which bacteria? That is gram-positive bacteria. What is its cell wall containing? 60 to 70% of mucopolypeptide, 70 to 80% of mucopolypeptide, 80 to 90, 90 to 100%. Right? So here, cell wall is made up of a mucopolypeptide, of course, but what is the percentage of that one is very important one here. So it is about 70 to 80% of mucopolypeptide it contains. Right? Is the right answer for this question. Next one. Lamb-brush chromosomes are found in. Already you know what are lamb-brush chromosomes, where they are available. So everything about lamb-brush chromosomes, they are a kind of giant chromosomes, you know it better. Right? Let us see the options over here. So salivary gland of mosquito. So salivary gland in which usually polytene chromosomes are available. Right? So next one, oocyte of amphibians. Of course, it is present in the oocyte of amphibian. Already I have mentioned in the theory part. Right? So that's why oocyte of amphibians is the correct answer for this one. And the next, silk gland of the silkworm, none of the above. Right? So this is usually, lamb-brush chromosome is present usually in the oocyte of amphibians. Next question. Cellular recognition and adhesion are facilitated by components of plasma membrane. These components are generally what? What are these components actually? He is asking. So you know better that there are some proteins are embedded inside the plasma membrane. So which is lipid bilayer one, and non-polar fatty acid tails are also present over here. You can observe this one. Yes. So this is how the plasma membrane is. Along with this one, some of the extrinsic proteins are also available over here. On those proteins, some of the carbohydrate chains, they are attached over here. So these are forming. If these are attached with the proteins, then it makes the glycoprotein. Right? So if they are attached with the lipids, then it is glycolipids. They are called as glycolipids. They are the cell recognizers actually. Right? So then let us see now. So which are the options given over here? Components of plasma membrane. These components are generally protein molecule alone. Lonely proteins are not cell recognizers, they are acting as lipid molecules only. No, only not the lipid molecules, both lipid and proteins. Right? He says both lipids as well as proteins. No, but whenever these form the glycoproteins as well as glycolipids, when they combine with the lipids as well as the proteins, so they are the cell recognizers. Then the answer for this question is glycolipids as well as glycoproteins is the correct answer for this question. Next question. In germinating seeds, fatty acids are degraded exclusively in the, right? So within the germinating seeds, fatty acids are degraded over here. Especially in what structure they are degraded. In germinating seeds, there are some cells are there. Within those cells, some of the cell organelles are there. Those cell organelles in which the fatty acids are degraded. So then which are those cell organelles? Especially they are microbodies. What they are? Microbodies like peroxisome, glyoxisome, mitochondria as well as proplastids, he is saying. So proplastids are the So precursors of plastids, right? So they are later become plastids. Proplastids later, so become plastids also. But here these are not the answers. So next one, mitochondria, he says it is also not the microbody, right? In which, so the fatty acids are degraded over here. So they are the glyoxysomes. They are none. They are the glyoxysomes in which fatty acids are degraded in germinating seeds. So that's why glyoxysome is the answer. So in theory we have studied this one. Next one is peroxisome is also not. Right? There the metabolism of peroxide that take place, formation of peroxide that take place in peroxisomes. Next question is, export form of body is. So this is the cell organelle which help in exportation of some of the materials, right? So and that is nothing but the cell organelle which help in packaging and so sending them off those materials out of that cell organelle. So those are especially the Golgi bodies of the Golgi complex. So is the correct answer for this question. That's why export form of the body is Golgi complex. Is the correct answer for this question. Not the endoplasmic reticulum, nucleus or mitochondria. Right? So next question is so very important one. The telomeres of eukaryotic chromosomes consist of short sequences of telomeres, especially they are consisting of short sequences of some of the nitrogen bases like. So these are nothing but nitrogen bases they are. They are the components of the DNA itself. DNA itself is a sequence of, so these nitrogen bases are usually it is having, right? So that is why, so you don't know all these nitrogen bases and how they are arranged in the DNA. So you don't know, but still he has asked over here the question. So it is a higher studies you will see in a PUC 2 in detailed manner regarding this one. All these nitrogen bases as well as chemical nature of the DNA. So in the next chapter also we are going to learn about the structure of this DNA, right? There you will come to know all these one. But remember in telomere of eukaryotic chromosome, there the majority is what is that guanine rich repeats are usually present. That's why. So the answer for this one is so guanine rich repeats. Okay. Next question is, yes, the next question is which one of the following is correct in plant cell? Which statement is correct in plant cell? That is the thing what we are studying now. So bigger vacuole with rigid cell wall. Of course, bigger vacuole will be there with rigid cell wall will be there in plant cell. It is a correct statement. So next statement is centrioles take part in cell division. No, no centrioles are present in the cells of the higher plants. Centrosomes are inactive and non-dividing cells, right? So centrosome itself is consisting of a pair of centrioles. Then if centrioles are absent, automatically centrosome is also absent there. Absence of cell membrane. Of course, in plant cell membrane is compulsorily present over there. That's why the first statement itself is the correct statement over here. That is bigger vacuole with rigid cell wall is the correct statement. Next question. Spirosomes contain. What spirosomes contain? Mainly they are meant especially for the storage of the lipids especially. So here, so we will glance over all the four options. Let us identify the correct one. Cellulose reserves. No, it doesn't contain any cellulose. Protein reserve. Of course, it contains protein as well as fats. So then which is the correct answer for this one? Both protein and lipids is the correct answer for this question. He says protein reserve here, he said lipid reserve here. So lipid is maximum, protein is minimum, but both are present in the same microbody, that is spirosome. That's why option D is the correct answer for this question. Next question. Site of gluconeogenesis is. So first of all, we must know what is gluconeogenesis. See here glucose is present. This glucose is formed from breakdown of the polysugar, right? So it is formed from the polysugars. Of course, it is quite natural. Polysaccharides undergo division. So means breakdown of polysaccharides into simpler molecules, that is glucose molecule, that take place, right? It is quite natural. But what happens over here? The synthesis of this glucose take place from non-carbohydrate materials. So sugars are nothing but carbohydrates. So here carbohydrate, that is in form of polysaccharides, it breaks to form monosaccharides as well as oligosaccharides, like this. So disaccharides, like this. Among them glucose is formed. It is quite natural, but so the formation of the same glucose that take place from non-carbohydrate component. Non-carbohydrate components like proteins, they can convert sometimes into glucose, and even fats, they can convert themselves into glucose. So that is the process which is called as what? It is called as gluconeogenesis. So it is the process which is taking place in which of the microbody? Gluconeogenesis that take place especially in Lysosomes, right? Where it is taking place? In Lysosomes. It doesn't take place in Golgi bodies, mitochondria, and none of these. So that's why a correct answer for this one is Lysosomes. Is the correct answer for this one. Next question. L-shaped chromosomes. L-shaped chromosomes I have already explained regarding this one. L-shaped chromosomes are also called as acrocentric. He says acrocentric. Means the centromere is at the terminal part. Say for example, if it is chromosome, this is the chromosome called as acrocentric chromosome. If second option, telocentric chromosome. See here, so if the centromere is present at the tip of the terminal part of the chromosome, then it is a telocentric chromosome. Next option, submetacentric chromosome. Submetacentric means the centromere is present a little away from the center. So it is submetacentric chromosome. See, if it is bent, then how it looks like. See here, if it is bent, then it looks like L. Hence, L-shaped chromosome is also known as submetacentric chromosome. Is the right answer for this question. So, I hope there is no doubt at all for you people. I have clarified all these things in the theory part only. Right? So, next question we shall go. Cytoskeleton is made up of. What is cytoskeleton made up of? Microtubules it is made up of. Microfibrils it is made up of. So, intermediate filaments it is made up of. All these three together form the cytoskeletal system. Right? Then, what are the options? Let us see and clarify the answer over there ourselves. Option A, callose deposits. No callose deposition is seen over there. Cellulosic microfibrils. One thing, cellulosic microfibrils, he says. It is not cellulosic microfibrils. Calcium carbonate granules. No, calcium carbonate granules do not make these one. They are microtubules, especially tubulin protein they are made up of. Means here protein is required over here. That is proteinaceous filaments. So the skeletal system of the cell, that is cytoskeleton, is made up of proteinaceous filaments. So there are three types of proteinaceous filaments already I have mentioned. One is microtubule, microfilaments, as well as intermediate filaments. That's why correct answer is proteinaceous filaments. Next question, very important question it is. The thickness of unit membrane is, already I have shown this one, the thickness of unit membrane. So how thicker it is, alright. So it is called as unit membrane. Why? Because it is not made up of a single layer, it is made up of 3 layers. Hence it is named as a trilaminar model of plasma membrane. See here you come across a lipid bilayer, and next one is non-polar fatty acid tails, here polar lipid heads, and those lipids are especially phospholipids. You know the aspects, right? So lipid bilayer. So since it is trilaminar, this is one layer, this is another layer. This is the lipid layer, bilayer, and the middle one is also there. It is consisting of, it is a light layer, since it consisting of non-polar fatty acid tails itself. So this one is 20 angstrom and this one is also 20 angstrom in thickness. I mean lipid bilayer, each layer is 20 angstrom in thickness, and the middle light layer itself is 35 angstrom. Totally 20 plus 20, 40 plus 30, 70, 70 plus 5, that is 75 angstrom that is in its thickness. That's why the correct correct answer for this question is option A, that is 75 angstrom. Alright, so this is the correct answer for this question. Next question we shall go. What is the next question? During endocytosis, what happens here? What is endocytosis? Already diagrammatically I have shown you. So directly we'll go to the options and search for the correct option over here. So the cell divides its cytoplasm during mitosis. He says cell divides its cytoplasm during mitosis. It is said as endocytosis. It is called as no. Then what is it is called as? There are 2 steps that we come across during cell division. What are those 2 stages? One is nuclear division. That nuclear division itself is called as karyokinases. And second one is cytokinases. What is the cytokinases? Cytokinases in the sense that cell cytoplasm division itself, right? After nuclear division, it is followed by the cytokinases. Of course, endocytosis he has given one. Cell digest itself. No, cell digest itself is not the correct answer. The cell engulfs internal materials using its membrane. The cell engulfs internal materials using its membrane. The cell enables an extracellular digestion of larger molecules. So then which is the correct answer for this question? See here, endocytosis in the sense that the internal materials have been digested over here. That's why the correct answer for this question is option C. Option C. Next question is, in sodium potassium pump of active transport there is, see here, active transportation, I had given example. Here there are some pumps that we come across. They are the sodium pumps as well as potassium pumps. See here, in an antipode system what happens over here is, suppose this is the membrane. This is the membrane. So the membrane protein is there, transmembrane protein is there. Here this is inner side of the cell, means cytoplasmic side. This is outer side of the cell. Consider. So what happens over here? From inner to outer, 3 sodium ions have been pumped out. Simultaneously 2 potassium ions that come in. So this is called as the antipode system. The movement of the molecules in opposite direction, right, due to vary in the concentration gradients. So, and that's true with the loss of energy, right? So it takes place over here, right? So whenever this antipode system that occurs over here, so there the efflux of the sodium ions that take place. Efflux means ejection of the ions out of the cell, right? And next one is influx of the potassium ions. Influx means pushing inside the cell. Here the correct option for this question is, see here once again the question, in sodium potassium pump of active transport there is efflux of sodium. There are 3 sodium ions that came outside the cell and influx of the potassium, 2 entered in. That's why, so this is the correct answer for this question. Okay, efflux in the sense that ejection of the molecule outside the cell. So influx in the sense that taking in the ions. Okay, so then next question. Ingestion of solid or liquid substances into the cell is referred to as, ingestion of some solids or liquid materials. So one is the amoeba that we consider over here. This is one single cell. What happens? It engulfs the solid food material, right? The solid food material is engulfed by this amoeba-like organism, right? And the liquid food, it may be solid or it may be liquid. So if any one of themselves is intaken, then the condition is said to be as phagocytosis as well as pinocytosis. Pinocytosis, phagocytosis, you know it better. Here what are the options? Let us glance over. Why? Because all the options would not be here. So what happens? Let us see. So it is ingestion of solid by liquid substances into the cell is referred to as diffusion, he says, exocytosis, he says, endocytosis, he says, osmosis, he says. Actually it is called as endocytosis. Here what happens over here is, so here just intaking of the solid or liquid material that takes place and intracellular digestion of that material takes place, right? So this is endocytosis, it is called as. That's why correct answer for this question is endocytosis. What is mytoplast? This I haven't covered in theory. See here, during the study of mitochondria, we must concentrate on the membranes that surround the mitochondria. See here, it is a double membrane bound cell organelle. It is very clearly known fact to you people, right? So during drawing the diagram of this mitochondria, you will find what this is one mitochondria. It is double membrane bound cell organelle, right? So this is outer membrane, this is inner membrane. Outer membrane as well as inner membrane that we come across here. Sometimes what happens? Simply removal of the outer membrane is being done. When it is removed, the mitochondria itself is called as mitoplast. Hence, the correct answer for this question is membraneless mitochondria, he says. Mitochondria will not be membraneless at any cost. So, second option, mitochondria without inner membrane, he says. Inner membrane must be there, right? Even it can lose the outer membrane, but inner membrane shouldn't. Another name of mitochondria? No, it is not at all the another name of mitochondria. Mitochondria without outer membrane is the correct answer for this question. Mitochondria, option D. Mitochondria without outer membrane is called as mitoplast. It is called as mitoplast. Is it clear? So next question I shall go over here. So what is the next question? Let us see. In animal cells, lipid-like steroidal hormones are synthesized in. See here, it is one of the functions of one of the cell organelles. What is the major function? Here, in animal cells, lipid-like steroidal hormones means the synthesis of a steroidal hormone is the major function of what? So see the options there. I have glanced over or I have given you in theory the functions of almost all the cell organelles, and there you can observe yourselves. So the synthesis of steroidal hormones that take place usually in endoplasmic reticulum. That's true. There are two types of endoplasmic reticulum that we come across. One is called as rough endoplasmic reticulum and second one is called as smooth endoplasmic reticulum. But the question is, in which type of endoplasmic reticulum these steroidal hormones are synthesized? The best example for steroidal hormones are, so there the androgens of the male, which is responsible for secondary sexual characters in male, like testosterone is steroidal hormone, right? Progesterone, right? Estrogen. These are all steroidal hormones that are respectively produced in males as well as females. It's a fact. Such kind of steroidal hormones are usually formed in one of the cell organelles called as smooth endoplasmic reticulum. That's why answer for this question is smooth endoplasmic reticulum. You will see that one in notes. Next one. Example of an oxidative organelle. It's quite a unique feature of the cell organelle. Here one of the microbodies is performing oxidation process, right? Oxidation process has been done by one of the cell organelle, and that is nothing but oxidation that take place inside the peroxisomes, right? So that is why the answer for this question is peroxisome. Right? Peroxisome is the answer. Oxidation that takes place to form the succinic acid from the glycolic acid. Right? Hence, peroxisome is the correct answer for this question. Next question. See here, which one of the following is not a cell inclusion? See here, many cellular inclusions that we come across, they are present inside the cytoplasm itself, right? So there may be some of the fat droplets are there, starch is also there, some of the crystal materials are also there, but one more is vacuole. Vacuole is not cellular inclusion. Vacuole is one of the component of endomembrane system. It is one of the cell organelle, right? So that is why the correct answer for this question is vacuole is the correct answer for this question, right? Now I hope you are very much clear with all these aspects. So whatever the theoretical aspects that we have studied in theory, on that basis only all these questions have been solved by me, right? So, and apart from all these questions, many more questions are there with you people in your material. So you go through all those questions and they are easier enough, right? I hope you will solve all other questions so before me itself, right? So it is possible from you people, right? Totally maximum number of questions I have solved over here, and while solving these bits, if you have any confusions, if you have any doubts, keep your notes with you people and see the notes and clarify the answers. Okay, so majority of the time, so it is possible to get the answers from the notes itself. So some of the questions which are little bit more than NCERT, right? So as to know them, it is better to go for reference. So reference itself is notes, right? And our Tungal material has elaborated knowledge is so there inside the material. So you refer that one and solve all those bits, right? So immediately you solve, and so before me once again you have to try to solve the bits. Whenever the theory classes are going on, that moment also you go through all these questions, then it will be very easier for you people to crack the answers quickly every day, right? And so next, so some other questions I will solve, I will try to solve, and after that one I will go to the next chapter, that is cell division and cell cycle and cell division chapter. It is related to this cell only. That's why first of all I will finish that one and will solve majority of the questions there also, and after that one we will go to the next topic. Thank you.